You're listening to an Airwave Media Podcast.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Steier
and Dr. Sarah Scheinman.
And if you tuned into last week's episode, which we hope you did, we had the Dr. Paul Offit on the pod. We talked about lots of things, but one of the things we focused on was some of the communication missteps early in the pandemic, particularly when the COVID-19 vaccines first became available. And this week, we are joined by the Dr. Michael Mina, still having a pinch me moment, to talk about why the COVID-19 vaccine never could have actually stopped transmission and why we were sort of, I don't know, I hate to say it, but like doomed at the onset with some of our messaging and how that really has led to a lot of mistrust around vaccines in general. And then we're going to have a whole other conversation about how this relates to the measles vaccine. Obviously, there's lots of talk right now about MMR. We could talk about boosters and titers and all the things because Dr. Mina is an expert on all these things. And I think it'll make for a great conversation. Yeah. So we should also tell you that we had every technical issue under the sun before recording this. So I am joining from my phone. I hope that it has not affected the quality of the recording too much. But Sarah is going to read Dr. Mina's bio today.
For a change. All right, everyone. Dr. Michael Mina, MD, PhD, is an epidemiologist, immunologist, and physician whose work bridges engineering, mathematics, and biology to advance public health. His research spans synthetic biology for immune monitoring, the population impacts of vaccines and infections, and diagnostic testing and mathematics for epidemic threats. He regularly advises U.S. and international health agencies on vaccines, immunity, and public health strategy. Dr. Mina was previously an assistant professor at the Harvard Chan School of Public Health and Harvard Medical School and associate medical director of molecular virology at Brigham and Women's Hospital. He also led the Broad Institute COVID-19 testing platform, which enabled widespread PCR testing during the pandemic. He led the Biden administration's home test to treat program and worked with the administration and NIH to create and lead the COVID-19 rebound cohort study to evaluate risk of rebound following antiviral treatment. He is also the founder of the Immune Observatory, a non-for-profit bringing next-generation immunomics to public health. He now serves as a fractional chief scientific or medical officer and scientific advisor across biotech and life science companies, from startups to Fortune 10 companies. He collaborates across academia and supports government public health policy. Wow.
Whoa. Woo. Wait, but two things. First of all, Dr. Mina, it must be like torture to sit through this bio that you're having read about you. But also, we really want to articulate your unbelievable expertise and experience, which is obviously very relevant to our conversation.
Seriously, we actually tried to cut it down a little bit, but we kept being like, no, that's important. That's important. The folks need to know about this. They need to, so we really... Yeah, we're so grateful to have you.
And Sarah, also, I have to just say, there's a lot of pressure reading bios, right? Because you're like,
My God, I have so much more respect for you now for doing that. Woo!
Thank you. And one thing I would be remiss if I didn't say, and we were just joking about this before we hit record, that there are so many times you read words, but you never say them out loud. And it's actually Broad Institute.
Broad?
It's spelled broad, but it's Broad. So I just wanted to give that.
Pardon me.
There you go. Well, Dr. Mina, we're going to call Michael on this episode. He's given us permission. Thanks so much for joining us.
Well, absolutely. Very happy to be here.
All right. So there's a lot of ground that we want to cover, but could we start with this conversation of could the COVID-19 vaccines ever have halted transmission?
Diving right in.
Yeah, seriously. I would say No, and I think I was probably one of the earlier people to really question that part of what we were seeing. And there's really good biology to understand why. It's not something that a lot of people study or talk about, but in order to really have a fully sterilizing or even partially sterilizing vaccine, meaning that if the virus lands, it doesn't get to replicate at all. It gets destroyed by the immune system before it even gets started. To really have that type of vaccine would require, for something like SARS-CoV-2, the life cycle of the virus just isn't right, if you will, for a vaccine, especially something that's not like a nasal vaccine, to be able to produce enough immune products, to actually coat the upper respiratory tract epithelial cells and mucosa enough to actually prevent something like SARS-CoV-2 from landing, replicating, and going away. And by that, I mean transmitting. And so what we saw early on when the vaccines were first tested and the data was first described out to the public, we did see early signals that maybe the vaccines were stopping transmission. The problem with those early signals is that they were all measured, and the whole vaccine trials were all measured at a time and a duration when the actual immune response to the vaccine is absolutely peaking. And by that, when you get a vaccine and you get an immune system response to the vaccine, your body in response also to infections, so this is kind of across the board, infections and vaccines, in response to that, your body creates an enormous number of what we call plasmablasts. And plasmablasts are just there to tackle the virus, in real time. So they expand. It's part of your B cell compartment. They pump out huge amounts of antibody, tons of antibody. And that antibody is able to, there's so much of it that actually exudes out of your gums and into your mucosa, and it leaves the vessels, it leaves your blood. Those antibodies are IgG antibodies. They leave your blood and they actually get into the mucosal surfaces. And during that period of time, you do have enough antibodies actually to generally at least depress transmission, because the virus lands, it gets gobbled up, neutralized by those antibodies, and it can't go on. But the problem with putting too much weight on those early studies is that plasmablasts, the cells pumping out these antibodies, are programmed to die. Mm-hmm. They're not long-lived cells. They're literally programmed to die within a couple of months or weeks, weeks or months after you get that vaccine. They start to fall away by definition, because they're just there to tackle the immediate threat and fall away. A fraction of them go and become really long-lived memory cells. But by and large, the antibodies wane. It doesn't mean your protection against severe disease wanes, and we can get into that. But the important thing is just the massive abundance and that burst of antibodies that happens falls away. And, this is immunology 101 in a lot of ways. So the fact that we had the New York Times talk about miscommunication across the board, people were surprised that antibodies were waning against the vaccine or the infection. It's nobody should be surprised, because that is what happens, especially in those early first exposures to a pathogen. And so what we saw was of course, the plasmablast died as anticipated. And with that death, the antibodies fell down over the next month or two in individuals. And then once you get 4 or 5 months out from a vaccine, its ability to really prevent transmission for a virus like SARS-CoV-2 is already markedly diminished relative to those first 2 months after vaccination. And that was kind of step one of not recognizing that aspect of the biology that I think was really damaging to our ability to communicate appropriately what people should expect from their vaccines, because that's a normal, natural process for a totally successful vaccine. We just put too much pressure on the vaccine, if you will, or we communicate. And by we, I like to think that I was trying not to communicate that. I was one of the people trying to say, hey, we should use more caution. Actually, a lot of epidemiologists, my colleagues, kind of railed against me for cautioning in early tweets when the first Moderna and Pfizer studies came out. There's a tweet that got shared quite a bit and then was kind of resurfaced more recently, where I said, like, these results are great, even for efficacy, but I would really caution, because the duration that we've actually studied it so far is just within that. Like, the trials were just like 3 months post. We only evaluated the vaccines for the first 3 months. And so studies after that really looked at transmission effects over those first few months post vaccination. And so it led to a lot of kind of heightened enthusiasm about what these vaccines were really capable of doing. And we haven't even gotten into the importance of mutations in all of this, and Omicron, and how expected, at least for me, that was another thing that I was arguing for early. And a lot of my colleagues said, you don't have any evidence for that. It's like, well, this is a brand new virus. Of course it's going to mutate. It's like, this hasn't been with us for a century.
Okay, I didn't know we were almost like the bad boy of epidemiology. I love it. Okay, so just to sort of recap for our listeners, we're talking about how early in the pandemic, I'm sure we all remember that the messaging I remember was, you get the vaccine, you won't get COVID.
Yes, that was definitely what was circulating. Yeah.
Oh, yeah. CDC, I mean, yeah, absolutely. That was the message.
And now we've pivoted and we're saying what you said, which is the vaccines are really good at preventing severe illness, hospitalization, and death. Yeah. That's the goal of the vaccines. But it's very different from our messaging early on. And now we have all these people messaging us, well, you told us originally this, and why did you change your mind? And so there's been a reversal in messaging, and that's what's led to a lot of mistrust. And so you're saying in very technical, but like, this is the necessary language, right? That there is something about the virus that made it that the vaccine could not have halted transmission and prevented us from getting infected.
Yeah.
Nope. What did I get wrong? Correct me.
No. I want to say it's not. It is about the virus, and it's about this type of virus. So one of the other pieces that goes along with this is, like, I hold that this is SARS-CoV-2 is still very much a textbook virus. Another thing that people railed on me for saying early on, but it's not. I want people to know that this isn't like unique to COVID, to SARS-CoV-2. This is upper respiratory virus infections in general. It's why we see people get many infections, over and over throughout lifetime. Unlike measles, polio, smallpox, there's different types of viruses, and it's the life cycle and how viruses replicate in our body that really, and how that interplays with our immune system, that is driving this. So you're right, it is about the virus, but it's not unique to COVID.
So here's a question then, like, why is it that some vaccines are so effective at preventing the illness altogether, but other vaccines are really, like the COVID vaccine, are really good at just preventing severe infection or hospitalizations, but not necessarily the infection itself?
Yeah, so the short answer is different viruses have different patterns and ways in which they infect the body. So let's just, like, COVID, like flu, are, SARS-CoV-2 and influenza virus are they have some of like the sort of simpler infection dynamics. They land on the respiratory epithelium. The cells of our respiratory tract are actually the cells that they replicate in, and then they go away, and then they transmit onwards. So that's very fast. It's an acute respiratory virus infection. Land, replicate, go. So just the speed of it and the access to the very cells that they need to replicate inside of, being right there on the same cell, being the same cells that they land on initially upon infecting the body, means that in order to prevent that process from happening, again, you'd have to have huge amounts of antibodies and immune cells kind of standing at the ready. It's kind of like saying, imagine if you had an army battling, a kingdom. And the kingdom had, big brick walls and you couldn't get into that kingdom. But let's say you had one type of enemy when their whole goal is just to, like, attach themselves to the brick wall. That's the whole goal. And for them, and like maybe there's some weird zombie things, and once they attach themselves, then they can, like, cause damage. I don't know. Well, it's going to be really, really, really hard to have enough soldiers there all the time to prevent enemies from just touching the exterior wall of the kingdom. Because the wall is there to actually prevent them from getting in. So if all they need to do is like touch that wall, well, that's really hard. It takes a lot of soldiers all the time to be there. If you have a different type of virus, like measles, which actually does its replication inside the body more. It's not the cells that it lands on. In fact, measles and other viruses where the immune system and the vaccines against them actually like totally stop transmission and really kind of symptomatic infection altogether. Those viruses tend to be viruses that don't just land here, replicate and go. They actually land and they get pulled into the body through dendritic cells, like these sentinel cells that are constantly sensing new stuff. And they pull them into the body. And then it's actually where the replication occurs is internal, if you will. If for measles, it's inside the lymphoid organs. It's inside of our immune cells themselves that measles has to replicate. And that process of having to like kind of dive all the way into the body first, replicate inside the body, and then eventually grow to such high amounts inside the body that then it like bursts out and transmits onward. That internal replication cycle of a measles virus is actually where it has just tremendous opportunity to bump into a B cell or a T cell that recognizes it. And so even small amounts of B and T cells inside the body can go a very far way to stop symptomatic disease of measles, because just the chances of them coming in contact are very high, because measles has to like go into the body. It has to like circulate through all these cells. And if a B or T cell inside there recognizes it, well, boom, it goes into its mode of expanding. That one cell might become billions of cells, all tackling that measles virus. And so it's really about the life cycle of the virus. The viruses that have to dive in can be stopped from transmitting onwards because the replication cycle is blunted by during that internal replication phase. A virus like influenza, rhinovirus, coronavirus, other beta coronaviruses, they just land, replicate, and go. And so it's just much, much harder. So going back to that like defense, the kingdom thing, it's the difference between having to like really get in and kill the king to take over the kingdom versus, just hitting the brick wall and being like, okay, our job is done. And so those are really different. The requirement is really different, and the ability to protect the king or queen or something, it doesn't take nearly as many forces to do that if you have like good layers of protection.
Okay. Oh, sorry, Sarah, were you going to say something?
I was just going to say, I love all of the analogies about the immune system so much. Like everyone who talks about the immune system has a slightly different analogy. And I love it. I love them all.
So we have to talk MMR, we have to talk measles. But before we do, there's a lot of chatter about, why do we need this annual update to the COVID vaccine, similar to the flu vaccines? There's talk about the development of a universal vaccine. Like, could we ever have a vaccine where we get one vaccine and we're protected for life against flu, we're protected for life against COVID? And I guess when we say protected for life, we're talking about from getting super sick and dying from those things. Like, is that feasible?
Well, for certain viruses, obviously it is, like measles. So what we say for measles, for example, even though there's multiple genotypes, meaning that there's multiple variants of measles, we say that there's one serotype, meaning the antibody and B and T cell and plasma cell response, the immune memory response against the measles vaccine is actually somewhat universal against all the measles variants that are out there. And that's because, in part, it's because it's a whole virus vaccine. Our immune system is educated around all parts of the virus when you get that vaccine, not just one little protein that's likely to mutate. And so with flu mutates enough that even with live virus vaccines, even with whole virus vaccines, the mutations in the hemagglutinin in particular, the protein that actually flu uses to bind to the cells, it mutates enough in what we call drift. It drifts away and mutates that we don't seem to have a universal vaccine, especially not for multiple lineages of the virus. Now, I would argue that the vaccine does do a good job universally, if you will, with the exception of brand new lineages and pandemics, to prevent severe disease for flu. But that's not now that we're kind of like up here. So when we measure the marginal benefits of seasonal flu vaccines, we're already here for our protection. And so the marginal gains are marginal. Mm-hmm. And sometimes they're losses, so this is kind of how fragile that targeting of the right virus and the right kind of freezing that drift and saying, okay, this is the one we're going to vaccinate people with. It's quite fragile. But the important thing for people to know is that we're already starting at a pretty good level. If people had no immunity to flu, which nobody in the world has no immunity except maybe the youngest among us who've never had a flu vaccine or an infection, then we would, if somebody got a flu infection for the first time as an adult and had never gotten vaccinated or infected, they'd probably get pretty sick, kind of like what we saw with COVID. And so we do in some ways have, sort of universal coverage in terms of some level of protection and quite a lot of protection against influenza. It's just that we're actually asking something pretty extraordinary of these vaccines. We're actually asking them to stop infection. And based on the last conversation we just had, it's pretty darn hard for a virus like this, for an immune system to possibly maintain that number of troops at the line on the battlefield to really prevent fully prevent infection. So I do think that, on the one hand, we sort of have it, but on the other hand, like we don't have sterilizing immunity. We don't have a universal vaccine that prevents all infections. Will we ever get there with something like flu? I think it would be hard. The closest thing, it's not so much about the type of vaccine we're giving. It's almost more the location and where we can get those immune cells to form. So this is why folks like those at Yale and others have really been working on, well, can we make nasal spray vaccines, intranasal vaccines, where they're actually driving long-term memory cells to exist right here in the respiratory mucosa. Will that work? Are they as long-term as like a plasma cell, which lasts decades? Tough to say, but is it possible maybe to really engineer a vaccine so that it's actually producing the antibody products, the antibodies themselves, the immune products in the right location to actually create more of a sterilizing kind of environment.
So one quick note on that term sterilizing. I remember, I guess it was 2020, 2021, I don't know, there was a lot of controversy in the SciComm world about that word because some people were unfortunately taking that to mean that the vaccines were causing sterilization.
Yeah. Oh, God.
Right? That we, right. Oh, yeah. That was a very big thing.
Really?
Oh, yeah.
Oh, no.
So just to clarify, are you, and this is my non-immunologist brain, so are you saying stopping the virus from replicating? Is that what we're talking about when we talk about sterilizing immunity?
That's right. So the sterilizing immunity is generally if you have let's say you have a coat, let's say your epithelium is totally coated with antibodies against that virus. The virus binds those antibodies, just glom onto it, and before it even has a chance, it can't replicate. It's gone. It's destroyed. That's sterilizing immunity. There's another term, and I think, because you are so focused on communication and helping people to understand and recognize where communication has failed. There's a few terms like this that I think really, I'm glad you call it out, that, behoove us as public health experts to recognize when we're saying things that the community just isn't, it's just not grabbing on. And so there's another term that we call herd immunity or community immunity. And man, did this drove people, it led to so many hardships in explaining vaccines and transmission issues, both for measles and for COVID. People, when I would talk about herd immunity and whether we would ever achieve it, whether it's for measles or COVID or whatever, you always get a whole faction of people who say, Dr. Mina doesn't know what he's talking about. He actually believes that giving a vaccine to somebody over here is going to somehow create airborne transmission of immunity to their neighbor. And, it's a valid point to question, like, if they don't understand what we mean, because herd immunity doesn't even make sense. In some ways, it's almost like us using the term. We're actually, it's not a good term, but it's an example. And the reason I say that is because herd immunity is really about transmission blockade. We should be calling them like transmission blocking vaccines rather than herd immunity inducing vaccines or whatever. There's got to be better terms, and there are better terms, is the point. And I always hold that as an example to remind me of, people have different vocabularies. And if we're speaking in an academic conference of epidemiologists, great. But when I go on CNN, I probably shouldn't say herd immunity because that just makes people think I'm like, that actually scares people more. Like now people are thinking immunity is airborne and vaccines must be doing something really crazy. It's right. So it's really an interesting topic, I think.
I mean, especially with this term sterilization too. I mean, because there are certain terms that mean one thing in the context of science and biology, and then another thing entirely in the context of like general knowledge and the public. And my favorite example of this is sugar.
Yep. Yeah, you're absolutely right. And even amongst epidemiologists, actually, there are some terms that are, I don't want to say controversial, but like seasonality. I've said very much that SARS-CoV-2 is a seasonal virus.
Right.
And, by that, I'm speaking much more on like how we think about seasonality as some predicted periodicity that we could like mathematically model with some function. That we call, it's not like, I'm not talking about winter, summer, fall, spring. I'm thinking of it more like, well, you have baseball seasons, which aren't like based on the weather. It's like, well, it's a baseball season.
Right.
It's when you play baseball, and the point is, even there, when I say that I believe very firmly that SARS-CoV-2 is seasonal, some people take that to mean that, well, then out of season, we shouldn't see any transmission. I don't think that that's true at all. I just think that there's an excess contribution in terms of seasonality in some periods of time. Like as an example, we see the peak in wastewater samples across America happen like almost within like the same week every year in January, in many northeastern locations, for example. So clearly there's seasonal components to this, even if it transmits in the summer. Well, even that has actually become quite predictable. But other people will say, no, this isn't seasonal because it transmits in the summer. It's like, well, it's just different ways of thinking about seasonality, even amongst infectious disease professionals.
Another example that came up on the conversation with Dr. Offit, and something that a brilliant immunologist on our team, Dr. Amy Pugh Bernard, feels very strongly about, is natural immunity. And it's because, the public, there's this huge appeal to nature fallacy. Everything natural is better. So certainly natural immunity is better than vaccine-induced immunity, right? And so I think Dr. Offit, what did he say, Sarah? We should call it survivor immunity or something else to really shift the language there. But maybe this is a good bridge to the conversation about measles, which you know a lot about. And there's this, I don't know, have you seen this? People keep bringing up old Brady Bunch episodes. You've seen this, right, Dr.
I have not. What's going on with the Brady Bunch?
Oh, yeah. So and maybe you can be more specific than I can, but I guess old episodes of Brady Bunch sort of portrayed it as like, no biggie, like, you had the measles, it's just a rash. Like, we sort of a very casual conversation about measles. So why are we suddenly so up in arms about measles and talking so much about the MMR vaccine? And it just blows my mind knowing what we know about how death is certainly not the only outcome of measles, and how there's this immune amnesia and all these other terrible potential things that could happen if you are naturally infected by the virus. So I don't know, maybe can you just, with everything you know, Michael, like what would you want the public to know about measles? And then maybe we could talk about something that we've already sort of had a discussion about. We've done some content on this about how the MMR vaccine is so unbelievably effective, and actually even likely more effective than we think it is.
Sure. Yeah. So the interesting thing with measles in that Brady Bunch episode is we have to put it in context. When at that time, measles did infect effectively 100% of all humans. But that doesn't mean that it wasn't harmful and it wasn't killing people. What it does mean is that it was normalized because it literally infected every single person when they were kids. So of course, it was, people didn't live in dread of it because it was bound to happen. So that's also why they had things like pox parties, which the measles, the Brady Bunch episode is about. That doesn't mean that measles was not bad and was not killing kids. The reason they had pox parties was because parents intuited and knew that in the pre-vaccine era, measles was inevitable. And so the question is, when do you get it? It wasn't if you get it. It wasn't how do I avoid it? It was when am I going to get it? And actually, if you get it later on in life, it can be worse. So that's why people had pox parties, because it was literally almost assured to be inevitable. If you lived like in a rural farm, maybe you'd get to 18 and never get measles. But you know what happened? Then you got recruited into the army and you became, so you were like a rural farmer in Kansas. And this was real. These are real studies, and real descriptions of these events. You'd have all these young men who would have survived all the way to 18, never got measles because they were in a rural area. So there was nobody around them to give them measles. They get recruited into the army. They show up in the barracks, and now you have a whole cluster of unvaccinated, because this is pre-vaccine, previously uninfected individuals who never got measles. And then boom, you have these outbreaks in the barracks, and they're devastating. Huge numbers of deaths. The death rate in from measles of these young guys who were, 18, 19, 17 years old, was extraordinary. And so it's not surprising that the mantra, probably rightly so, was, hey, get measles a little bit earlier. Right. And so it drove this idea of like, okay, we got to get pox parties, but it doesn't mean that kids were not dying of measles. We know that the death rate is, depending on sort of your baseline nutrition, baseline health, is between 1 in 100 cases can die to 1 in 1,000. That's huge. It's in kids, it's literally orders of magnitude worse than COVID. And so I wanted to read something because you had, and I happen to have this on I keep it on my desktop because it's, it's a New York Times article from 1963. So if we want to know how people actually felt about measles, like, let's just go back in time and like, don't try to recreate the history. Like, let's just go back to articles that were published in 1963. That's when the vaccine came out for measles. And like, it's a few paragraphs long, but I'll just read one or two sentences. It says, 2 types of measles, the vaccine against measles have now been adjudged by the United States government authorities as acceptable, essentially for use in the U.S. This will come as welcome news to the parents of young children not yet exposed to measles, particularly of those between the ages of 6 months and 5 years for whom the disease is most dangerous. And then it goes on and it says the most dreaded complication is encephalitis and inflammation of the brain and the spinal cord that may lead to permanent damage. And then it goes on to talk about, the death rate and the number of deaths as compared to polio. The point is, this is how people were actually thinking about measles before they were privileged enough through vaccination to forget what measles does and the ravage it has. And so, I think it's very, very important to keep that in mind. I'll stop there because you asked a few questions, but I just want to, if you have thoughts, I'm happy to talk about, which way do we want to go on that?
Yeah. I mean, we always say public health is a victim of its own success. And that's the perfect example of that. So now we have this amazing vaccine. I just remember, when I was a grad student, however many, 15, 20 years ago, I remember it was always 97% of, that was the stat that we were fed. But you did, there was this great, was it on Twitter or Threads or something, like a very back of the envelope calculation that showed, well, actually, if we use the data of this current outbreak that originated in West Texas, it's actually looking like it's something closer to 99-ish% effective. Yep. Can you just talk a little bit about that?
Yeah, absolutely. And then I want to come back to it and mention immune amnesia and some of these other interesting effects. But certainly, so, the vaccine, generally is, considered to be 97% effective. CDC, WHO, everyone kind of says that, Well, it occurs to me, could we, is that the right number? If it's 97% effective, then what that means is that during an outbreak, 1 in 30 people should get measles, who are vaccinated, which is actually, because it's 3%, 3% of the population who are vaccinated should be susceptible.
Right.
So 1 in 30 is kind of a big number if you're vaccinated and walking around in a community like the community in Texas, to think that 1 in 30 people should get measles who have been vaccinated. But it's not what we're seeing is the important thing. So while we quote 90%, 97% effective at stopping measles, that might sound really high, and it is really high already. So the good thing here is we're already starting at an amazing baseline. And what I'm saying is maybe it's actually quite a bit higher. Maybe it's not 1 in 30 people might get measles upon exposure. Maybe it's more like 1 in 500, which is a big difference. It doesn't sound that much to say 97 versus 99.5. It all sounds like high numbers.
Right.
But actually you're getting into almost another order of magnitude protection, 1 in 30 versus 1 in 500. And so the simple back of the envelope calculation to do here is just to say, well, okay, we have, let's say in Texas, something like 600 or 700, I'm not sure the actual number right now, individuals with measles who have been infected. And we know that almost all of them are unvaccinated. We know that about 30 of them have been vaccinated. So you get 30 infections. So if you were just calculating, okay, that's 30, out of 600, that sounds like a lot, but that's not the right calculation. It's more like 30 out of all the number of people who've been exposed,
right?
Which is easily thousands and thousands of people. And probably if we assume that there's significant underreporting of measles cases, it's probably now over 1,000, maybe even 2,000 actual measles cases that have happened, measles infections that have happened. And we don't expect massive underreporting of vaccinated individuals who do get measles. Probably a lot of people who have been vaccinated who start to get measles will probably go and get a test and say, what is this? And so given those kind of numbers, when we only have 30 or maybe it's even less than that, true known vaccinated cases of measles happening, it actually points to us, the numbers actually show that it's more like 99.5, 99.7% effective to stop infection and transmission, which is remarkable when you think about, the fact that this is 2 doses of a vaccine given when people are 1 and 3 years old. And then at least people born in the late 70s and beyond have more or less never been exposed and naturally boosted. It's amazing to realize how much these vaccines really do drive very strong immunological memory that lasts us, a lifetime so far as we can see.
And also when we're talking about it doesn't sound like 97 versus 99.5. It's both an A.
Potato.
Yeah. It's both an A. But when you're thinking about these are actual people, it's very easy when we're talking about large numbers and population-based statistics to kind of lose track of the fact that these are individuals, these are children, these are folks that are really getting sick and dying. So that few percentage points translates to, lots and lots of real people.
Yeah.
That's right.
So and I know we're coming close to the end of the episode, but 2 things. One, I know you wanted to talk about immune amnesia. We should definitely talk about that. I feel like we just casually mentioned that. That's something that more people need to understand what we're talking about. But 2, you said something just now about how the protection lasts us a lifetime, right? But there are a lot of people who are concerned, what if my protection waned? And there's talk of, should I get titers checked? Should I get another dose of the MMR vaccine? On our team, we've put out, primarily in conjunction with Dr. David Higgins, who's a pediatrician and epidemiologist, Dr. Amy Pugh Bernard, who's an immunologist. And they basically said, getting titers checked, just skip that step because it doesn't capture all of the other, all the types of protection that and I'm butchering this, but basically it's checking for antibodies, but it doesn't capture things like, what is it? Memory,
B and T cells. That's right. Yeah.
Immunity. Yeah, exactly. All kinds of other stuff. And so they're saying, like, if you're concerned, if you're not sure about your, whether you got both doses, you could safely get another vaccine. But really, if you received both doses of the MMR vaccine as a child, you can safely assume that you have protection. So what say you?
You're absolutely right. And I think that's, it's a little bit of what I was just saying, what we were just talking about. One in 500, if it's 99.5, 99.7% effective, that's across all people, and this isn't like some reevaluation of some old data. This is like just doing the back of the envelope calculation of what we're seeing today.
Right.
And it even accounts for things like clustering and all, this is actually like kind of a low ball estimate. It's like 99.2% would be like the lowest we would go based on what we're seeing today. And what that means is that most people should feel very confident that if they were vaccinated and had 2 doses of vaccine, that they are protected today. Do I recommend people go run out and get their titers checked? I don't. I don't recommend that. Some hospitals, I was boosted because I was kind of forced to be boosted because I had to get my titers checked when I started working at Brigham and Women's Hospital. My titers were below what they deemed the protective threshold. And that triggered me having to get a booster. I argued a little bit, but then I didn't have the energy to care very much. I said, okay, fine, I'll get the booster.
Pick your battles.
Yeah. But to Dr. Higgins' point, antibodies are just one part of our immune repertoire. And so what we were discussing before, where measles actually has to like dive into the body before it can replicate and transmit and cause disease. Even a small number of B and T cells, even if like your plasmablasts, your plasma cells that live in your bone marrow are real like long-term antibody producing immune memory actually exists in our bone marrow. So even if that's kind of like stopped pumping out antibodies as much, which is what the antibody titers are checking, you might have B and T cells that are floating around. And even a small number of those can actually be fully protective against infection because, again, the virus has to like circulate all through inside the body. And if a B or a T cell like recognizes that virus, then it goes through this massive exponential expansion. And like 1 or 2 cells that recognize that virus can turn into billions. And so boom, like that, you have a good response. So that's why we're seeing just this massive efficacy of the vaccine even decades later. Now, would I tell somebody who's really concerned and wants to know not to get a titer? Like, absolutely not. Go get a titer. And you know what? If it's low, get a booster if you want. I actually believe if I had to place a bet right now, I bet that as we move into like maybe 15, 20 years from now, maybe even less, maybe 10 years, I wouldn't be surprised if we do start to see some breakthrough infections at a higher rate amongst people who have never been naturally exposed, but only vaccinated. And why I say that is like, we're only now getting to the first time in human history where we're getting individuals getting to a slightly older age group who have never been infected and exposed over and over and over. Even people in the 60s, after they got vaccinated, measles was still spreading. So even people who got a vaccine in the 60s and therefore never got measles probably actually were exposed to measles quite a few times just out in the world. So we're only now getting to people born in like the early 80s who literally have never been naturally exposed. And so who are their entire immune memory and immune protection is based on those 2 vaccines. And so as we get older and those people get into their 60s, that's when your immune system starts to kind of deteriorate a bit. And so for some people, we might actually see a higher rate of breakthroughs. And so I wouldn't be surprised if we do see a recommendation come about sometime in the next decade or 2 for individuals in their 50s or early 60s to get an MMR booster because of this.
As an 80s baby, I'm taking notes.
Right.
All right, poor Sarah, I know you have to get to the lab. Maybe just a final thought from Michael about just this immune amnesia. Just I feel like it would be so powerful coming from you. Like, What is this, and why should we not take measles lightly?
Yeah. So the short of it is there's a tremendous amount of damage the virus does to our body. When measles infects the body, it can lead to encephalitis and, infection of the brain that could actually cause death. 10 years later, the virus can actually pop back out and cause a panencephalitis that kills you. It's 100% lethal. Happens in, 1 in a few thousand cases. So it's not super prevalent, but that's not prevalent either. The 1 in a few thousand is very high. To just, know that if your kid gets measles today, in 10 years, their brain might just start deteriorating and they die. It's 100% fatal. So that's one thing. But it causes respiratory failure. It causes bacterial co-infections. It causes acute immune suppression. So these are all things that happen during the acute phase of the virus that drive hospitalizations and deaths. And it's worth saying that a measles infection will put in around 1 in 5 kids will end up being hospitalized. Around 1 in 100 to 1 in 1,000 will die. 1 in a few thousand to 1 in 10,000 will get this encephalitis 10 years later. But then there's a more insidious phenomena called immune amnesia, which is something I helped to discover. And that was in two science papers. And
you discovered this? Okay, I didn't know that. How we? Oh. Okay, keep going.
So those were 2 papers in 2015 and 2019 that I first authored in Science to describe this phenomena. And what it is and I want to be clear, it was with terrific colleagues at Princeton, Brian Grenfell and Jess Metcalf, and a terrific colleague and postdoc mentor at Harvard, Steve Elledge, and then the late Diane Griffin, who is an amazing scientist at Hopkins, and a great colleague, Rick de Swart, in the Netherlands. So those are the main folks working on this project. And what it is that when measles gets into the body, it actually destroys pre-existing immune memory. So all that good immune memory and immune protection that a kid develops in their, through struggling through colds and infections in their first year of life, and you have kids getting RSV and going to the hospital. So kids kind of go through this first few years, any parent knows, of like kind of tumultuous immune building. It's called getting sick as a little kid, but sometimes it can be risky and dangerous. And what happens when you get measles is all that immune memory that's built up during those first years of life can actually be erased. And it's because measles literally feeds on immune memory cells. The B cells we've been talking about, the T cells we've been talking about, the plasma cells we've been talking about. These are the 3 cells that hold our immune memory, kind of like our brain for our immune system. Mm-hmm. And the crazy thing about measles is it binds specifically to immune memory cells. Its receptor is highly expressed on immune memory cells. So if you think about a receptor like a lock and key, a virus needs to have a key, and different cell types have the lock. For COVID, it was ACE2, which is it's like on a lot of cells in the body. For measles, it's specifically a molecule called CD150 that lives and exists and is expressed on immune memory cells. So when measles infects the body, it's not just going in and infecting like all of your cells of your body. It's specifically replicating and killing your immune memory. And that drives this long-term loss of immunological protection that you then have to like regain. And we've shown that it takes years to potentially rebuild that immune memory.
I love that we're just like cramming this in at the very end of the episode. We're like, yada, yada, yada.
Yeah, we need to do a whole nother episode.
No, I mean, my goodness, that should be a pretty convincing reason for folks to get that vaccine. I still don't understand how we're talking about this, but anyway, well, Michael, this was unbelievable. I feel like we really could talk for hours. There are so many things I know we wanted to talk about, we just didn't get to. So maybe we could entice you to come back on the pod at some point. But thank you so much, just for everything you're doing and also for joining us today.
Well, very happy to have joined you and always happy to come back.
Yeah. Thanks so much, Michael. And thank you all for tuning in. We hope you learned a thing or two. Please join us next week on your trusted source for no nonsense, just science.